Genome sequencing. Sequencing is the first step of structural and functional analysis of newly determined sequence. The importance of sequencing is increasing to find the interface between new protein and the one which already exists. DNA sequencing commonly refers to the methods using to determine the order of the nucleotide bases in DNA. Biological researchers are totally dependent on the knowledge of DNA and are accelerating day by day. Other research areas in which sequencing is making use of are diagnostic, biotechnology, forensic biology and biological systematics. Sequencing started in 1953 by the establishment of DNA double helix by James Watson. The next sequencing was RNA sequencing of first complete gene and genome of bacteriophage MS2 by Walter Fierce in 1972 and 1976 respectively. The large gap between the first two sequencing is because different DNA molecules hold similar chemical properties that made it difficult to separate them. The chain length of naturally occurring DNA molecules was much greater than for proteins that made complete sequencing unapproachable. The existence of only 4 bases in DNA made the sequencing difficult for DNA than for proteins. The DNA sequencing was very difficult without the knowledge of DNases. But after 1970, there were several achievements in DNA sequencing. In 1973, Walter Gilbert and Alan Maxim reported the sequence of 24 base pairs using a method known as wandering spot analysis at Harvard. In 1977, Frederick Sanger developed a sequencing method at the MRC Center, Cambridge. The complete DNA sequence of the Epstein-Barr virus with 170,000 base pairs long was sequenced by the scientists of Medical Research Council in 1984. Then, the first semi-automated DNA sequencing machine was developed by Leroy E. Hood's laboratory at the California Institute of Technology in 1986. In 1990, large-scale sequencing trials on Mycoplasma capricolum, Escherichia coli, Caenorhabditis elegans and Saccharomyces cerevisiae has started by US National Institute of Health at a rate of $0.75 per base. The first complete genome of free-living organisms, Haemophilus influenzae, the bacterium, is sequenced by Hamilton Smith and colleagues at the Institute for Genomic Research in 1995. Since 1990s, the usage of sequencing methods has increased day by day. The sequencing methods that are discussed here are 1. The Sanger method and its most important variants, 2. The Maxim and Gilbert method and other chemical methods, 3. The pyrosequencing method, DNA sequencing in real time by the detection of released pyrophosphate, and 4. Single molecule sequencing with exonuclease. Exonuclease digestion of a single molecule composed of a single strand of fluorescently labeled deoxynucleotides. Maxim-Gilbert sequencing. This is a method of DNA sequencing developed by Alan Maxim and Walter Gilbert in 1977. This method is based on nucleobase specific partial chemical modification of DNA and subsequent cleavage of the DNA backbone at sites adjacent to the modified nucleotides. This method is coming under the first generation DNA sequencing. This is a chemical cleavage method using double-stranded DNA samples and involves modification of bases in the DNA followed by chemical base specific cleavage. The base specific cut occurs at a small proportion of either both purines, that is adenine and guanine, or preferentially at A, or the pyrimidine, that is cytosine or thymine, or at cytosine only. The resulting DNA fragments are then denatured, run side by side in slab gel electrophoresis, autoradiographed and analyzed. Purified DNA can be read directly. Homopolymeric DNA runs are sequenced as efficiently as heterogeneous DNA sequences. The method can be used to analyze DNA protein interactions, which is a part of DNA footprinting. Also used to analyze nucleic acid structure and epigenetic modifications to DNA are some of the advantages of this method. But this method is not so commonly used these days because it requires extensive use of hazardous chemicals. It has a relatively complex setup, technical complexity. It is difficult to scale up and cannot be used to analyze more than 500 base pairs at a time. The read length decreases from incomplete cleavage reactions, and it is difficult to make Maxim Gilbert sequencing based DNA kits. Initially, all the steps of these chemical sequencing methods were performed manually. Years later, a system composed of a computer controlled microchemical robot that carries out of one of the 4 reactions, that is guanine, adenine plus cytosine, cytosine plus thymine or cytosine alone, in less than 2 hours was described. The key points in the Maxim Gilbert methods are the chemical reactions. They can be separated into 2 different groups. 1. 4-lane methods where 4 or more separate cleavage procedures are used and the information is displayed in 4 or more parallel gel lanes, and 1-lane or 2-lane method where all reactions are based on only one chemical modification and electrophoresis is performed in a single or 2 lanes. The first report of a single lane method was based on a chemical cleavage procedure that uses hot aqueous piperidine for several hours. In 1991, Negri et al. Described a 2-lane method which can become 1 lane by mixing the products of the 2 reactions, where the labeled DNA fragments is heated in the presence of formamide. The result is an efficient cleavage of the phosphodiester bond at the 3' residues adenine, cytosine and guanine with the relative efficiency A is equal to G greater than C. The bias between A and G is obtained through pre-treatment with methylene blue. In conclusion, the main advantages of the Maxim Gilbert when compared with methods are: 1. A fragment can be sequenced from the original DNA fragment instead of from enzymatic copies. 2. No subcloning and no PCR reactions are required. Consequently, for the location of rare bases, the chemical cleavage analysis cannot be replaced by the dideoxynucleotide terminator method, as the latter analyzes the DNA of interest via its complementary sequence. It can thus only give sequence information in terms of the 4 canonical bases. 3. This method is less susceptible to mistakes with regard to sequencing of secondary structures or enzymatic mistakes. Despite of all these advantages, most of the protocols have some drawbacks. First, the chemical reactions of the protocols are slow and the use of hazardous chemicals requires special handling care. The worst problem is the occurrence of incomplete reactions that decreases the read lengths. Sanger's method or chain termination method. The first method described by Sanger and Colson for DNA sequencing was called plus and minus. This method used Escherichia coli DNA polymerase I and bacteriophage T4 DNA polymerase with different limiting nucleoside triphosphates. Ionophoresis on acrylamide gels has been used to resolve the products that are generated by the polymerases. Due to the inefficacy of the plus and minus method, Sanger and his co-workers described a new breakthrough method for sequencing oligonucleotides via enzymatic polymerization. This method was initially known as the chain termination method or the dideoxynucleotide method. It consisted of a catalyzed enzymatic reaction that polymerizes the DNA fragments complementary to the template DNA of interest. Briefly, a P32 labeled primer was annealed to a specific known region on the template DNA, which is a starting point for DNA synthesis. Catalytic polymerization of deoxynucleoside triphosphates onto the DNA is occurred with the help of DNA polymerases. The polymerization was extended until the enzyme incorporated with a modified nucleoside known as terminator or dideoxynucleoside triphosphate or DDNTP into the growing chain. This method was performed in 4 different tubes, each containing the appropriate amount of one of the 4 terminators. All the generated fragments had the same 5' end, whereas the 3' end was determined by the dideoxynucleotide used in the reaction. After all the 4 reactions, the mixture of DNA fragments with different size was resolved by electrophoresis on a denaturing polyacrylamide gel in 4 parallel lanes. The pattern of bands showed the distribution of the termination in the synthesized strand of DNA, and the unknown sequence could be read by autoradiography. In this figure, you can see the various steps in Sanger sequencing method. A single stranded DNA template, a DNA primer, a DNA polymerase are involved. To each reaction, only one of the 4 dideoxynucleotides such as DDATP, DDGTP, DDCTP or DDTTP are added. Following rounds of template DNA extension from the bound primer, the resulting DNA fragments are heat denatured and separated by size using gel electrophoresis. The base, that is at the end of each strand, has a fluorescent tag attached, a different color for each basis. These colors can be seen with a fluorescent microscope, and the sequence of the DNA is determined by the sequence of colors that come up on the gel. A computer reads the colors and determines the sequence, which it then combines with all other sequences to make up the whole genome. Now, we can study the different approaches in Sanger sequencing method. 1. Direct approach. In direct approach, the sequencing of unknown DNA is performed within the sites in which the sequence is known. The unknown DNA sequence is amplified by inserting it into a vector. The first sequencing reaction is performed using the primers that hybridize to the vector sequence and polymerize the strand that is complementary to the template. Then the second priming site has been chosen in the newly generated sequence by following the same direction as the first reaction. This is known as primer walking, and it helps to reduce the redundancy. However, it requires the synthesis of each new primer, which in the past was time consuming and expensive, especially when dye labeled primers were used. Nowadays, the appeal of a cost effective and time saving method that uses small oligonucleotide libraries has disappeared with improvements in primer synthesis technology. However, the demand for a sequencing method that was able to provide long read length, short analysis time, low cost and high accuracy has led to several modifications of the original Sanger method. In addition to several improvements in the procedures and in the reagents used in the sequencing reaction, further development in DNA separation technology was of paramount importance for the completion of the human genome project. The next method is di-terminator sequencing. Improvements in the DNA polymerase enzyme increase the quality of the sequencing reactions and data. Isothermal DNA polymerases were used at the beginning for manual and automated sequencing. The reactions were performed at 37 degrees Celsius for 20 minutes. That helps the T4 and T7 polymerases to incorporate with all 4 terminators. But these polymerases are very sensitive to temperature that can easily deactivate it by a temperature fluctuation. With the discovery of the polymerase chain reaction and the use of a heat stable DNA polymerase called Taq polymerase, it was possible to perform sequencing reactions with reduced amounts of DNA template compared to isothermal enzymes. In this approach, dideoxynucleic acids or DDNTPs are used instead of deoxynucleic acids. Although the approach is largely improved, there was still significant variation in peak intensity for fluorescently labeled dye terminators. The pattern of the termination was reproducible and predictable, but this variation made automatic base calling difficult. A few years later, one of the major suppliers of fluorescent sequencing kits introduced a modified set of fluorescent labels for DDNTPs. With this new dye terminator kit, the signal was more even and automated base calling improved significantly. The next method is automation and sample preparation. Automated DNA sequencing instruments or DNA sequencers can do 24 runs a day with 384 DNA samples in run. Size separation, detection and recording of dye fluorescence are performed using electrophoresis, and data output is getting as fluorescent peak trace chromatograms. A number of commercial and non-commercial software packages can trim low quality DNA traces automatically. These programs score the quality of each peak and remove low quality base peaks, which are generally located at the ends of the sequence. An example for the output file is shown in the figure. In short, Sanger sequencing provides a high degree of accuracy, long read capabilities and the flexibility to support a diverse range of applications in many research areas. Sanger sequencing is highly recognized for DNA sequencing applications, but also supports applications in RNA sequencing and epigenetic analysis. Also in Sanger sequencing, each individual reaction is fairly cheap and very accurate.